Good afternoon. Yes, I'm working as a scientific officer for an NGO, for the NGO Doctors Against Animal Experiments that is located in Cologne in Germany. I'm a biochemist from background, and I've been working as a research group leader for a very long time on the development of human-based in vitro methods. The debate that we have today is not only an ethical debate, but animal experiments also need to be banned for scientific reasons. As we already heard, this is very important because animal experiments from a scientific point of view are not valid, they are not safe, and they are even a risk for human health. So they need to be banned. If animal experiments are banned, it is very important, of course, that we have alternative methods. And we need human-based methods because human-based methods produce human-relevant data. Alternative methods is actually not the appropriate term in this respect because these human-based models that already exist are not an alternative to animal testing because they are not as good as animal tests, they are much better over animal tests, they are relevant to humans and excellent research tools that we already have. So today I want to show you that these models are already existing and that they are used very successfully for research, also on cosmetics and on toxicology purposes. Our organization, Doctors Against Animal Experiments, is a charitable organization which was founded in 1979. It consists of around 3,000 members, of which 1/3 are scientists or doctors and scientists working in the medical field. We provide scientific information against animal experiments for scientists and also for the broad public. We organize campaigns, actions, lectures, and also scientific congresses, etc. Our main aims are the abolition of all animal experiments for ethical and primarily for scientific reasons. Because, as I already said, animals are not a good model for studying human-related issues. And therefore, we also call for an increased support of animal-free research, including human-based methods that can be used for cosmetics research. Animal tests for cosmetics have already been restricted in many places in the world, as you can see on the map here. And you can also see that in these places where they haven't been banned yet, there is growing public rejection. So this clearly shows that the debate that we are having today is extremely important. In the European Union, the testing and marketing ban for cosmetics exists since 2013, and this legislation has further driven the development of innovative animal-free research methods. However, these alternative methods have to be regulatory accepted, and the responsible institution is the EURL ECVAM, as we already heard. So this stands for EU Reference Laboratory for Alternatives to Animal Testing and European Centre for the Validation of Alternative Methods. The ECVAM is part of the Joint Research Centre, which is the Science and Knowledge Service of the European Commission. And the job of the ECVAM is conducting and coordinating research activities and validation studies for the safety assessment of chemicals. As you can see on the scheme, during the can you go back? As you can see on the scheme, during the validation process, the ECVAM is in close interaction with several cooperation partners and also a number of stakeholders. And the ECVAM also takes into account opinions from the SCCS, which is the Scientific Committee on Consumer Safety. It also takes into account testing guidelines, so methods that have already been accepted by the OECD, which is the Organization for Economic Cooperation and Development. And the validation process of the ECVAM can basically be subdivided into 4 steps. This is the assessment of the submitted test method. Then second, the planning and the conduct of validation studies. And this is followed by an independent scientific peer review by ESAC. And ESAC is the EURL ECVAM Scientific Advisory Committee. So this is an independent advisory committee. So after positive review by the ESAC, we have the development of EURL ECFAM recommendations, and finally the regulatory acceptance of the alternative method. Many testing procedures are important for cosmetics research in terms of toxicology and safety and risk assessment. So this is, for example, acute toxicity, dermal, oral, and inhalation route. It depends on the substance. That can be a fragrance, it can be a cream, and different other things. Hair dyes, for example, also. We have a number of toxicological applications. We have procedures for toxicokinetics, phototoxicity, genotoxicity, carcinogenicity, and then also things like eye irritation, dermal absorption, skin corrosion, skin irritation, and skin sensitization. So you see that there is a number of procedures that have to be conducted when a new ingredient of a cosmetic undergoes safety and risk assessment. The good news is that for all these procedures, already in vitro methods are existing. But there is always a but, of course. First of all, not all these in vitro methods are accepted as complete replacement of in vivo studies. For cosmetics, of course, this is the case because animal studies have been banned, but in other fields of toxicology, for example, it depends on the outcome of an in vitro study if an in vivo experiment needs to be added or not. Also, not all these assays are 100% animal-free. That means that some of them use animal-derived cell cultures or animal-derived additives, for example. Also, in vitro can mean ex vivo. So ex vivo means that for this assay, animal-derived tissue explants are used. First of all, this is ethically not acceptable, and again, we have the problem that animal-derived tissue explants are not a good model for human safety assessment. So there are already numerous validation and pre-validation studied in renowned scientific journals that definitely show that human-based, really animal-free models have a much, much improved quality compared to animal-based research methods. And some of these methods I want to introduce to you today, first of all, in the field of toxicokinetics. So how is toxicokinetics defined? Toxicokinetics is how the body handles a substance as a function of dose and time in terms of the concept of ADME. ADME is a very important term in the field of toxicology, and ADME stands for absorption, distribution, metabolism, and excretion. And this is the process how the substance to be tested is absorbed by the body, how it is distributed in the body, then how it is finally metabolized, and how the substance and metabolized are finally excreted. The in vivo animal experiments are mainly conducted in rats, but also in other rodents and other animals. The administration of the substance can occur via the oral, dermal, or inhalation route, depending on the cosmetic or on the substance. And then finally, urine and feces sample are analyzed, and finally the animals are killed because also tissues have to be analyzed. For oral administration, you have a process called oral gavage. This is what you see on the image here. Oral gavage is a very stressful procedure for the animals. And this is also reflected in the experimental outcomes. So if an animal is stressed by a procedure, the data and the experimental outcomes are not reliable. Also, this is something that has been proven by many, many scientific studies. There is a process also in toxicokinetics that is called repeated dose testing. That means that such an animal gets such an oral gavage, as you see here, for 28 or for 90 days every day. So in vitro test methods have been developed and have also been accepted by the ECFAM. This is, for example, for oral administration, the so-called CACO2, human intestinal cell line, which is a human-based method, and this cell line is derived from human intestine. For metabolism, the responsible organ is the liver. So here we have human primary hepatocytes, so liver cells that were isolated from humans, or the human HepRG cell line, also human-based liver cell line. For inhalation, We have no method that is currently accepted by the EORL ECFAM, but we have the MUSIL air system, which is currently under evaluation by the ECFAM. And this is a very complex model, highly innovative. So this is a complex 3D tissue model of the human airway epithelium. On the left side, you see a microscopic image, and you see that there are different cells, because every organ consists of different multiple cell types. And this is what you have in this model. And even the architecture, so you have different layers, like in the real lung. And even the system, as you can see, has little hairs, like you have in the human lung. So this is a reconstructed human airway epithelium, very close to physiology and very, very valid. The same models are also available for human skin, so we have reconstructed human skin models and reconstructed human intestinal models, for example. All these human three-dimensional tissue models are derived from human primary cells, which are obtained during routine surgeries from human donors. They are available from different human donors, not only from healthy, but also from people suffering from asthma, from smokers, or people suffering from COPD, which is the chronic obstructive pulmonary disease. And the impressive thing is that the phenotype of the human donor is conserved and reflected by the cultures. So if the donor is a smoker, then also the tissue culture that is generated is the phenotype of a smoker's lung. So also for medical research, this is very, very good model systems. And the good thing is also that these models are really completely animal-free. So even culturing of these models is animal-free. New substances for cosmetics also have to be tested for their eye irritation potential. The definition of eye irritation is changes in the eye following the application of a test substance. The in vivo test methods that has been really for a very long time has been used until I think 2009 is the so-called Drazy rabbit eye irritation test. So what is done is the substance is applied to the eye of a rabbit, or also other rodents, but mainly rabbits, and then the eye of the rabbit is observed for irritation. If the substance is irritant, this is a very harmful, very painful procedure for the animal. This is really a cruel experiment. And the animals are either killed after the test, if the substance was irritant. If it was not irritant, then the animal undergoes a certain so-called washout period and finally is reused for further experiments. It is the question which fate is better. And on the right side, you see that this test that has been used for such a long time is absolutely not valid and absolutely not reproducible. So this is a study, what you see here on the right side. And in this study, 3 different substances were tested by 24 different laboratories. So this is a so-called intra-laboratory trial. These 24 laboratories all had the same testing conditions, the same test substances, and what was measured is the so-called maximum average score, which is the endpoint measurement for the irritants of the substance. And as you can see, you can find every result that you want, beginning from 0, which means that some labs found that the substance is absolutely not irritant, up to a score of around 70, which means the substance is highly irritant. So this is the result of such a method. This is representative. This is not only the case for this test, but this is the problem of animal tests. So would you like to use a cosmetic that was tested in this way? Or also use medical products that were tested in tests with such kind of results? So this is really dangerous. Animal experiments are not only not safe, but they are also dangerous because they can harm humans, because the outcomes are not reliable and not transferable to humans anyway. Also for eye irritation, there are several in vitro test methods accepted by the ECFAM. Some of them are, as I already mentioned, ex vivo methods. So there is this isolated chicken eye test or the bovine corneal opacity and permeability test. These tests are also not the models of choice because even if they are in vitro models, they use animal eyes derived from animals. So also here we need other methods. There are also human-based, animal-free in vitro methods. that have been accepted by the ECFAM. This is, for example, the epiocular eye irritation test or the skinetic eye irritation test. Both of them are three-dimensional tissue models, like I showed you before, for the airway epithelium, for example. So we have here a reconstructed human ocular epithelium. And on the second image at the bottom, you see how such a cell system looks like. So these 3D tissue cultures are grown on a so-called cell culture plate. And here on this plate that you see, you have 24 identical ocular 3D cultures. So instead of 24 animals that need to suffer for a test, here you have 24 cultures. And then 24 different substances can be applied and tested. The test is very fast. The whole procedure takes around maybe several hours maximum. The measurement endpoint is tissue viability. So depending on the irritation potential of the substance, the tissue viability is good or less good, depending on how harmful the substance is to the culture. And then finally you measure the cultures in a photospectrometer, and then relative tissue viability is plotted against the substances, and as you can see on the last image, this is how it looks like, such an analysis, and you have a certain threshold. And if the tissue viability is above this threshold, this means tissue viability is high, so the substance was not irritant for the eye. And if the tissue viability is below this threshold, that means that the substance is declared as irritant. And this is a very, very precise and reliable method with a very high reproducibility. Skin corrosion and skin irritation are also parts of the safety and risk assessment in cosmetics research. So skin corrosion is defined as the irreversible damage of the skin, visible necrosis following the application of a test substance, while skin irritation is defined as a reversible local inflammatory reaction of the skin following the application of a substance. The in vivo animal test that is used or has been used for these purposes is the Drazy rabbit skin corrosion irritation test. On the left side, you see how this test works. So first of all, we have a patch application. So a patch is that contains the substance to be tested, is applied to a shaved skin area. And then in B, you see the skin of the rabbit at day 0, so when this test starts. In C, you see the skin of the rabbit at day 7, if the substance is not irritant or not corrosive. And then in D, you see the skin of the rabbit if the substance was irritant. So also this method is very harmful to the animal, and animals suffer from that. And as the eye irritation test, this method has been the gold standard test for a very long time. And also, here it has been proven that this method is highly variable. It has very, very limited reliability and very poor prediction of human skin reactions. So in vitro test methods for skin corrosion are also ex vivo methods. This is the rat transcutaneous electrical resistance test. And this test is also based on skin explants of rats, so also not the method of choice. And especially rat skin is well known to be a very poor model for human skin, because it is much thinner. It is like, I think, 10 times thinner than human skin, so absolutely not an appropriate model. Also, animal-free, human-based in vitro methods have been, meanwhile, accepted by the ECFAM, and this is all 3D tissue cultures of the human skin, so reconstructed human skin models. Like, for example, the epiderm or the epiderm FT model. These models are accepted as full replacement for the in vivo test. The regulatory accepted endpoint is also tissue viability, so the same procedure that I showed you before. And you can also measure inflammatory mediators that are released by the tissue cultures, which is important, especially in the context of skin irritation testing. The 3D human skin models, such as the reconstructed human epidermis models or the full-thickness skin models, very closely mimic the native human skin. These models are used for a number of applications also beyond safety and risk assessment. So for example, skin barrier function can be assessed, or skin hydration, also whitening and pigmentation, because these skin models are also available with different pigmentation. So with high, low, and medium pigmentation, depending on which ethnicity should be targeted. Also, skin aging, skin anti-aging can be tested, photo-aging, and UV protection. These models and also the other reconstructed tissue models have numerous advantages over animal studies and also over animal tissue explants. So first of all, no interspecies differences, because we definitely get human-relevant data from a human-based method. Long-term experiments are possible, which is not the case for tissue explants, even human tissue explants that are not cultured but just taken to be used in vitro, have a very limited viability, maybe a few hours, maximum 12 hours. These cultures here can be used over weeks. So you can really do long-term experiments and repeated application. This also leads to an excellent standardization of these models and reproducibility. You have personalized skin models because, as I already said, the phenotype of the human donor is conserved in the culture. These models are commercially available, they are validated, they are ready to use, and they are very easy to handle. And finally, they are time and cost effective. So while, for example, a skin irritation test with such an in vitro model takes a few hours, a test in an animal can take days or even weeks. So different 3D human tissue models that I showed you can be also combined and used to simulate, finally, a kind of human organism on a biochip. The so-called multi-organ chip technology, or organ-on-a-chip technology, has not been accepted for regulatory purposes yet by the ECFAM, but it is, for example, currently validated by the FDA in the US. And this is one of the most promising animal in vitro models, animal-free in vitro model, to be used for safety testing. An advanced model is the so-called ADME chip that you see here, which was designed specifically for toxicokinetic studies to measure absorption, distribution, and metabolism and excretion of a substance. So 4 different tissue cultures are placed on these chips, cultures of intestine, liver, skin, and kidney, and these cultures are combined or connected by a microperfusion system. And this microperfusion system simulates the blood circuit and the excretory circuit. And then you have, even within these circuits, you have different spots where you can do sampling. So you can kind of really take simulated blood samples and urine samples from this model. So there are different variants of these multi-organ chips, and different model systems can be used to simulate the human body. Very recently, for example, a 10-organ chip has been developed with 10 different organs on the chip. And it contains 3-dimensional mini-organs, such as stomach, liver, brain, and kidney, and many more. And these organs are generated using a highly innovative technology. The 3-dimensional mini-organs can even be grown from human hair root cells. So you don't need a tissue sample or a biopsy from a human being, but you can even generate them from hair root cells. These hair root cells are turned into induced pluripotent stem cells by genetic engineering in the lab. And then from these induced pluripotent stem cells, almost every organ of the human body can be generated in the lab. The size of the organs, of the mini-organs, is around 0.5 millimeter maybe, so they are very, very small. And then the organs, the mini-organs, are also personalized. So also these organs, like the tissue cultures, they conserve the characteristics of the human donor. Another approach to generate mini-organs and 3D tissue cultures of human origin is the so-called bioprinting technology. So this approach uses a 3D printer and bioinks that contain different kinds of human cells. And this enables the generation of very complex human-derived tissue cultures in a highly standardized manner. And also this throughput is excellent for, this assay is excellent for high throughput applications, which is important for industrial purposes, for example. Last but not least, we also have different kinds of computational models, so-called in silico models, that are used to predict the toxicity of substances and that have also impressive reliability. Some of these models have already been accepted for regulatory purposes by the ECFAM. This is, for example, the QSAR models. QSAR stands for Quantitative Structure-Activity Relationship. So the QSAR models predict biological or toxicological properties of a substance based on the physiochemical and structural properties. And you can have different endpoints, for example, toxicology, or also other endpoints can be addressed. Then there is the Pbk models, which is physiologically based kinetic models. These models are representing the ATME, so absorption, distribution, metabolism, and excretion of compounds in humans or animals. And these models enable an in vitro to in vivo extrapolation of toxicity data, and also the simulation of the internal concentrations of the substance in the body. And if you couple these models to target organ response models, so also take into account how the organs respond, then you have the so-called PBKD, so even more advanced models, physiologically based kinetic and dynamic models. So also here with computational methods, very, very good prediction is achieved. So what is the vision? The vision is, and this is also stated by the ECFAM, the vision is an integrated approach to testing and assessment, so the combination of all these human-based models for the assessment of, for the risk and safety assessment of chemicals. And animal experiments have to be banned because data from animal experiments are relevant to animals, and data from human-based models are relevant to humans. So for this reason, our aim must be the abolition of all animal experiments for ethical, for scientific, and also for safety reasons. And I really hope that I could convince you today that animal-free research is the science of today and of tomorrow. Thank you very much for your attention.